Hi everyone. So this is week 4 and it's our Intro to Spectrophotometry lab. It is also known as spectroscopy, and you might hear all about spectroscopy. It's a huge technique for biology, and you're going to be just introduced to the most important parts of it for biological purposes. In case you're wondering, I've got a broken arm. See, broken arm. So I have slowed down a little bit, but fortunately, spectroscopy is one of the things I know pretty well, so I can do a lot of this over video for you. And I might be a little slowed down in getting your grading back to you, but I can do a lot on dictation, so we'll see how it goes. For this lab, this week 4 lab will be due on February 15th, which is Monday, next Monday at midnight. It needs to be handed in one PDF only with your trinket on the first page. So photograph the first page with your trinket. If you can't print out at your home, you need to contact me via email and I will give you another procedure. But I need to know that ahead of time because you don't want to be doing this last minute. For this lab, you will be doing some graphing. So while in the scientific writing lab, you were talking about doing the graphing, now you're actually going to be doing the graphing, and it has to follow the CSE format. Give yourself time to make sure you get that right. All right. You have 3 activities in this lab. The first activity will be preparing a solution of methyl orange and recording the absorption spectra of methyl orange. The second activity will be creating a solution of bromocresol green and then recording the absorption spectrum of that. The third activity will be preparing a Beer's Law plot, which we'll go into, and determining an unknown concentration of cobalt chloride. Now you may be saying to yourself, how am I going to do this? I don't have any of those materials, and you don't need any of those materials. This is a simulated lab online. It's actually very effective for what we're trying to get across online. So you can do this all on an app. You'll use 2 different apps. They're quite good. And give yourself time to make the graphs for this. So you won't be able to race through this lab. Let's go through the highlights of this so that you can know what you're getting into. The background of spectrophotometry or spectroscopy is that each molecular compound has its own ability to interact or absorb wavelengths of light. Just like a fingerprint has a very unique set of grooves, molecules have their unique set of absorbing light, and that set is a known characteristic that you can compare and find unknowns by comparing it. You can identify compounds using spectroscopy, and you can also determine their concentrations using spectroscopy. We'll be exploring both. Let's understand a little bit more about these wavelengths of light. So you should be reading through this lab. I'm just going over the highlights of it. So in terms of that, in terms of light, light travels theoretically in packets of energy or particles of energy called photons. And light travels in waves. And light is not a particle and is not completely a wave. It's a hybrid of the two. I know that sounds tough to understand. Einstein figured it all out. So we kind of go with it. We don't know enough about light. If you'd like to explore it, the field is wide open. And what we do know is these particles of light that travel in waves have distances between their wave peaks and their wave troughs. These distances are very orderly, and in fact, the distance between the wave peaks is the exact same distance as between the wave troughs. So peak to peak is exactly the same distance as trough to trough. You can measure either one. They're identical, and that distance is known as the wavelength. These wavelengths are usually measured in nanometers, which is the symbol that you'll see here, nm. It's generally known as 10 to the negative ninth, nano-ninth. Interestingly enough, the longer the wavelength, the less energy of the light. In other words, the wavelength and the energy content are inversely related. Very important to remember. Inversely related. The longer the wavelength, the larger the distance of the wavelength, the lower the energy of the light. I teach astronomy and meteorology. We work with this all the time. So in other words, AM radio waves are very long waves. They can actually go and be as long and as tall as buildings and skyscrapers, which is why you don't get good AM reception in cities. It's a very long wave with very low energy. On the other hand, X-rays, very short waves, very high energy, to the point where you need a leaded apron, when I got my X-ray done, to protect the rest of your body from these very high energy waves that can change the DNA. They're very high energy waves and very short wavelengths for X-rays. The portion of the electromagnetic spectrum of waves from cosmic waves, even higher energy than X-rays, and AM radio waves, very low energy waves, that spectrum is called the electromagnetic spectrum. And the portion that we will study with our spectrophotometer is the visible side of that spectrum, or the visible spectrum of the visible section of that spectrum. It's called VIS, V-I-S, visible wavelengths. Those visible wavelengths go from 400 to 700 nanometers, generally. The 400 nanometers are smaller wavelengths. The 700 nanometers are larger wavelengths. And in fact, the smaller wavelengths right around 400 are generally blue light. Like ultraviolet is very high power. Then when you get into the blues, which are kind of like violet but not ultra, they're a little bit less powerful, but they're pretty close to ultraviolet. And what you know as infrared, under red, those are longer wavelengths like you see in heat bulbs. They're not really harmful at all. And that's the spectrum that you're looking at, from blue to red. In the middle is generally yellow, and then you have variations on all those themes to get the other colors. So we're going to be working in those wavelengths, but there are spectrophotometers that read other wavelengths. Those spectrophotometers can be exclusive for things like X-rays and gamma rays and cosmic rays, especially in astronomy that I teach. We look for cosmic rays and for gamma rays from far-off galaxies. And those other spectrometers could be useful for radio waves. All right, we're going to be working right in here. We're going to be working in the spectrum that goes from 400 nanometers to 700 nanometers. We talked about the shorter wavelengths, and, well, we'll talk about them a little bit more. Shorter wavelengths and longer wavelengths have different characteristics in the way they go through prisms. And Isaac Newton was the first to discover that when he put white sunlight through a prism, it came out the other side all separated into the colors of light. No one understood this before Sir Isaac Newton explained it. And he explained that the wavelengths of light are all mixed in to create the white light. A prism is another material that slows down the transmission of the wavelengths. They slow down inside the prism. When they slow down, they start to separate out. And when they separate out, the short wavelengths refract or change their angle more than the longer wavelengths. And that makes sense, because if you think about shorter wavelengths hitting something that's slowing them down, they're going to change their direction a lot more quickly than a big, long wavelength that hits something that slows it down. It's not going to change its angle nearly as much. So the red wavelengths, which are longer, refract less in a prism, and the blue wavelengths, which are shorter, refract more in a prism. Your spectrophotometer uses this knowledge to separate out these colors and determine what kind of a molecule or molecular compound you have. Let's explain that a bit more, but before we get to that, you can remember the order of diffraction by using the mnemonic Roy G. Biv, because red comes first, orange, yellow, then green, blue, indigo, and violet. All right? Roy G. Biv. Red, orange, yellow, green, blue, indigo, violet. Really helpful thing to remember from long to short wavelengths. When you are dealing with a spectrophotometer or a spectroscopy device, all of them have characteristic pieces to them. And those characteristic pieces are going to be a little bit different for each device, as is the software that's associated with them. But let's talk about the 5 basic parts. The 5 basic parts are the light source and the monochromometer, which we'll go into a little bit, is really the big system in the spectroscopy device. The sample holder, which holds your molecular compound. The phototube, which interprets what comes out of that compound in terms of light. And the photometric system, which records what comes out and starts to analyze it. It sounds like a lot, but a picture is worth 1,000 words. So I'm going to show you a picture. So you can look down at your page 5 and look through here, and it'll all come together. The light source is generally a light source that shows in this visible light spectroscopy device, it will show all wavelengths of light merged into a white light. So an incandescent light is a fine light source. From there, the light source goes through the monochronometer. And that monochronometer contains 2 things. It contains either a prism or a lens. And early spectroscopy devices all contain prisms. Now you have very specific lenses for most of them. They do the same thing. They slow down the light to break it up a little bit more into its wavelengths. Then the next part of your monochronometer after the prism is the slit, or the diffraction grating. The diffraction grating acts to pinpoint exactly what wavelength you want to go through your sample. In this case, this diffraction gradient has moved to pick up just the green wavelength of light. That green wavelength of light has its own energy. The energy is kind of in the middle. The violet light, which is pretty close to ultraviolet, is a pretty high energy visible light. The red light is a pretty low energy visible light. The green is just in the middle. This particular diffraction grating is a slit that moves up and down to meet the green light and only